So just a heads up, this is also being live streamed. And I have posted a final review on the Study Union WordPress that I would like to go over, if you guys haven't already done that. And if you printed it out like a day ago, I've made some changes to it, so it'd be best to use the current one right now. And you can find that by going to ucfstudyunion.wordpress.com. And the plan is we can go over the review that I've made, and then if we have any time left over, we can just go just like ask questions topic by topic, or if you guys want, we can do questions first and review. Doesn't really matter to me. And also, this review has not been made with the professor, any TAs. This is just me as an SLI leader making it for you guys, so it's just a heads up, right? So I guess we'll start off with number 2 first, right? Because it's like the most general question we have. It's pretty basic, right? So what does it mean to be diploid? 2N. Yeah. That's really good. So 2N. Does anyone else have anything to say about that? Thank you for that. 2 copies of the same chromosome, right? So we'll have homologous chromosomes, right? So a haploid individual would not have 2 copies of each chromosome from each parent, right? No maternal, no paternal, just one set of DNA, right? And so now back to number 1. Which DNA sequences are always identical? Is the answer D? So is the answer D? Would you guys agree with that? So would you say that sister chromatids and homologous pairs are always identical? Seeing some nods. Seeing some shakes. Okay, so we know could someone define for me what sister chromatids are? Yeah, awesome. Thank you for that. So yeah, sister chromatids, obviously we just didn't get baseline definitions down, right? Because this is pretty simple. I mean, not simple, but basic stuff, right? So you're going to get sister chromatids after S phase, right? So yeah, after S phase. And these are going to be genetically identical because they're held together by the same centromere, right? Because they're the same DNA chromosomes just held together, right? Then homologous pairs, are they genetically identical to each other? Not necessarily. Okay, so where is the variation in homologous pairs then? Are the loci in the same locations? Seeing some nods. Okay, yeah, so we have similar loci, but what is different about those loci? Alleles, right? We have different forms of genes, and then we call those alleles, right? So same location, but they're not the exact same sequence, so we're not going to say that they are always identical, right? So now number 3. Growth occurs during what phases of the cell cycle? Could anyone tell me the phases that growth occurs in?
G1.
Okay, G1, G2. Okay, awesome. Anything else? What about G0? That's a good one. No, there's no growth in G0, right? Because this is like what a cell enters when it's not dividing. So if, like, example, neurons, your neurons aren't really dividing, so they're not in the cell cycle. G0, you're not participating in the cell cycle. You're out of the cell cycle, right? So G1 and G2 are growth phases, right? I mean, G for growth. And S phase is obviously DNA replication. And the M phase is where our cell divides into two different cells. So number 4, a cell in the G2 phase has 16 chromosomes. In metaphase of mitosis, how many DNA molecules does this cell have? Okay. Hearing some 16s, 32s, okay. Thank you guys for that. So we have two different answers here, right? 16 and 32, and we're looking for the number of DNA molecules in the cell, right? So we're in G2, so that means we've done S phase, right? So we've replicated our DNA in our cell, right? And remember that these different representations, right? This is what a chromosome would look like before replication. And these are sister chromatids after S phase, right? And even though it looks like it's 2 chromosomes, right? This is still considered 1 chromosome. Right? So this would be 1 chromosome with 1 DNA molecule, and this would be 1 chromosome and 2 DNA molecules, right? So now we look back to the question. Our cell is in G2 phase and has 16 chromosomes, right? So that means we've had DNA replication. So now how many DNA molecules are in metaphase of mitosis, right? So if this is 2 DNA molecules and we're in mitosis, we have 16 chromosomes because this is 1 chromosome, right? So 32 chromosomes, right? And now let's say I changed metaphase to anaphase, right? And I asked you, how many chromosomes does the cell have? Be 32, right? I heard some 32s. Because in anaphase, these two are going to get pulled apart, right? So we still have the same number of DNA molecules within the cell, but our chromosome number has doubled because they've been pulled apart. And now there's 32 of these guys. So yeah, number 5 is basically what I just said. When do chromatids become chromosomes? What effect does this have on the number of chromosomes in a cell? I pretty much just said that. So obviously that's anaphase. That's when sister chromatids separate and become their own respective chromosomes, right? And for those of you that just walked in, I'm looking at my review that's on the WordPress, the study in WordPress. So number 6, how can we calculate the possible number of chromosome combinations? There's a good formula for this.
It's 2 raised to the n, where n is the number of chromosomes.
Awesome. Yeah, thank you for that. So she said it's 2 raised to the power of n, where n equals the number of homologous pairs, right? Because with the law of independent assortment, we know that these chromosomes are going to independently assort themselves on the metaphase plate and going to go into their own cell. So there's different combinations of chromosomes that can go into each of the different cells in meiosis. So during what phase does crossing over occur? All right, so I've heard some prophase 1s of meiosis. Seems like a pretty good answer to me. So what happens during crossing over? Does it generate new alleles? And what crosses over, right? Homologous pairs, sister chromatids, non-sister chromatids.
Crossing over between non-sister chromatids, and it doesn't generate new alleles, just variation among the alleles, only mutation between the alleles.
Awesome. Yeah, that's really good. Thank you for that. So yeah, note that crossing over is between homologous pairs, right? And these homologous pairs differ from each other. They have the same loci, but they have different versions of genes at these loci, right? So they're going to exchange their alleles to create new combinations of chromosomes that will then go into their gametes and create genetic variation, or not genetic variation, because genetic variation is caused by mutation, but it'll just reshuffle the deck of alleles, right? So now it's between non-sister chromatids, right? So what would happen if sister chromatids cross over? Yeah, nothing, right? Because they're genetically identical, so if we swapped two of the same cards, we would get the same cards, right? Could someone tell me what a chiasmata is? Chiasmata, area of connections between sister chromatids? Okay, thank you for that. Would you guys agree with that? Chiasmata? So what would we call the area where crossing over occurs? The what? Synapsis. Okay, that is involved in crossing over. Like that's pretty like synonymous to crossing over, right? You guys hear like synapsis crossing over, right? So if these are sister chromatids, you usually see like that representation in the center, and that's what the spindle microtubule is attached to pull the sister chromatids apart in anaphase, right? So what would we call this guy that holds the sister chromatids together? Centromere. Good. So would we say a chiasmata is an area of connection between sister chromatids? I wouldn't say that, no. So what do you guys think the answer would be? B? D? Thank you. Areas where crossing over occurs, I like that answer. That's a good answer. So now we'll look at number 10, right? When does the degradation of Chiasmata occur? So first, to answer this question, we need to know what Chiasmata is, right? So what's the function of Chiasmata? Protects the what? Protects the sister chromatids from separating? Keeps the chromosome together? Is that what you're saying? Cohesin from separating. Okay, awesome. Yeah, thank you for that. It's really good. Yeah, so Chiasmata is going to keep your sister chromatids together in anaphase 1, right? So anaphase 1, this is in meiosis, right? And as we know, we have cohesin holding together sister chromatids, or sorry. Yeah, so Shigoshin is degraded after meiosis 1, right? Because cohesin is the thing that degrades during crossing over to allow the homologous pairs to cross over, right? Because we have cohesin holding together these sister chromatids because we need the sister chromatids to stay together during mitosis or meiosis at the right times, because if these sister chromatids were to split apart at improper times during the cell division cycle, we could get some type of aneuploidy, right? And then one cell could accidentally get one of the sister chromatids that split apart when it wasn't supposed to. Then you get bad things from that, right?
So it's not after anaphase 2 because it happens at anaphase 2?
Yeah, that's a really good point. So, yes, you are correct. Because, yeah, just after meiosis 1 in general, right? Because we need Shigoshin to keep the sister chromatids together in anaphase 1, because we're going to have cohesin come in, or separase is going to break the bonds between cohesin in meiosis 1, so these two homologous pairs can cross over, right? Because if separase didn't break down cohesin for crossing over, these sister chromatids, they'd be stuck together, right? And we need these to overlap and exchange genetic information. So we need something to let these non-homologous pairs cross over, but if we didn't have Shigoshin, right, there'd be no protection for the chromosomes to completely stay together, right? So if we had no Shigoshin and then separase came in and broke down the cohesion in meiosis, it would just be a mess, right? Because we want this parts that are crossing over to be free and cross over, but we don't want the whole sister chromatid to break up yet, right? Because we know in meiosis 1, right, the homologous pairs are going to line up on the metaphase plate and split, right? Compared to mitosis, where the sister chromatids split in the first round, right? But meiosis 2 is where the sister chromatid splits, so meiosis 2 is pretty similar to mitosis, right? So 11. Is it B? B. Okay, yeah. So 11 is B, right? Meiosis 1 being a reductive division that's producing 2 haploid cells. And meiosis 2 is being an equational division producing 4 haploid cells, right? So what do we define an allele as? A? A form of gene found at a given locus. Okay. I like that answer. And the location of a gene along a chromosome would be called a locus, right? So now we'll look at 13. So a YO genotype is indicative of what? Lethality? B? Okay, lethality. Yeah, thank you for that. That's really good. So we're saying that YO is lethality, right? So which chromosome is probably the most important to have? X, right? Because, you know, in females, they still have 2 X's, but obviously the bar body is, the second X is going to turn into a bar body, and it's going to be, for the most part, inactive, right? So does anyone know what Klinefelter syndrome, what the genotype of Klinefelters? XXY? Okay, cool. Thank you for that. And triplo X is pretty self-explanatory, right? How about Turner syndrome? XL? Is that what you're saying?
Yeah, they don't have a second chromosome.
Okay. So Turner syndrome would be XO, right? Hi.
Random question. Do you have like a mic or anything you could wear?
I'm wearing one now, but I mean, it's obviously not like broadcasting. It's the mic's more for like the live stream. I can try to speak up. Yeah, okay, sorry about that. And who could tell me what super male condition would be? Super male. XYY, cool, right? So I mean, if your Y chromosome makes you male, then having two Ys will make you super male. Okay. So now, we'll look at number 14. So which organism in the XXXO sex-determining system is the homogametic sex? Female. Okay. And what would be the female in this case? XX. Okay, cool. So homogametic sex meaning they're producing one type of gamete, right? Gametic, gamete, homo, same, right? So what type of, if the XO is the male, what type of sperm is this male producing, right? If he's the heterogametic sex, he's going to be producing gametes of two different genotypes, right? So what's going to be in his gametes? X and nothing, right? So O is indicative of nothing. And 15 is just a simple definition question, right? Genic sex determination. B? Okay, thank you for that. So did you guys agree that genic sex determination is B? Do we have any other answers here? Genic sex determination? C. Okay, I like that answer, but I would maybe look down one. So D, right? So if let's just kind of like take the name literally, right? So we're having sex determination based on our genes, right? So genic sex determination. So in like human sex determination, we're going off of, I mean, it's still like technically genic, right? Because you're having the sex determining region Y on your Y chromosome, which is like a gene, right? So I mean, that still kind of is like based off a gene, but the difference is that males and females, we have like an actually different physical chromosome makeup, right? Like the Y chromosome's a lot smaller than females. You have like an X chromosome and a bar body, right? So in genic sex determination, the sexual phenotype is based on genes, but chromosomes appear to be the same across sexes, right? So now we will look at number 16. Hopefully you guys didn't forget how to do a problem like this. So I'll give you guys like a minute or two or three, work on it yourselves. We can discuss it. What's that? B. Hmm? 1 out of 32? Okay. So did you guys get, who here, did anyone get 1 out of 16? 1 out of 8? 1 out of 32? Nice. Okay. Does anyone not know how we got 1 out of 32? Anybody? Do you guys want me to explain it? Okay, we saw a hand. Okay. So when we're doing a cross like this, right? What is this, an and or type probability rule? It's an and, right? Because we want this offspring to have those A's and those B's and those C's and those D's and the E's and the F's, right? So obviously in and, we multiply the probabilities together. So you can do this a shorthand way by just looking at the parents and kind of figuring out what type of allele they're going to pass on to their offspring. Or you can actually draw out a Punnett square for each of these probabilities, right? Because obviously we know alleles or chromosomes segregate randomly, independently of sort. So these are independent events of each other. So we could just treat this as making a Punnett square of crossing. Like for the A's, we could just make a Punnett square, and we can just calculate that. Obviously, we would get, so then we don't really have to include that because it would just be the same thing. We divide our probabilities by 2, so you don't really need to put the second A in there. And we would get something like this. And then we look at, we need big A, and we have 2 possible probabilities. So it'd be 1/2 to get big A, right? And you just repeat the process over and over. That helps at all. That was very, very summarized for time's sake, but hi. Okay, yeah, so that is a no, right? So we're gonna look at like the probabilities of A, B, C, D, and F, right? So I just did A here, for example, right? And the probability that I would get a big A from these 2 parents if I cross both of their genotypes, let's say this is dad, let's say this is mom, it'd be 1/2, right? Because, yeah. So I would say A equals 1/2, right? Then I would go down the line and look at B's, C's, D's, and F's. Then I would multiply all of them together, right? Because it's back to the and rule of multiplication. Because and is meaning these events are independent of each other. So we're going to multiply their probabilities, their individual probabilities, to see what's the probability of me getting this for A and this for B and this for C, so on and so forth, right? Hello? Hello? Oh, that's loud. So now we'll look at 17. In a group of 30 turtles, 10 have no green pigment in their shells, 12 have a lot of pigment, and 5 have moderate amounts of pigment. 3 have very little pigment. So we got some turtles with a little bit of green, a lot of bit of green, no green. So what is this an example of? Variable expressivity. Okay, good. So what could we define expressivity as? Awesome. That's very good. So literally, right, the degree to how the phenotype is expressed. So we have a green phenotype. It could be not that green. It could be really green, kind of green. So it's just the degree which is expressed, right? And that's variable. So is that all we have here? Is this codominance? What's codominance? Two dominant traits showing up, right? So is it like a blend, or are they both like discrete? They're both there, right? So it's not a blend. So what is incomplete dominance then? In between, right? Or we could do incomplete intermediate, right? So if like dominance red, recessive white, maybe heterozygote would be pink, right? A little bit in between. So is this an example of variable penetrance? That's a good question. Oops. So to answer that question, we would need to know what penetrance means, right? Can anyone define penetrance?
How likely it is that if you have the allele, it's going to show.
Okay, right. So whether or not like we have the genotype, are we seeing the expected phenotype, right? So in this case, we have some turtles with no pigment, and we have some turtles with pigment, right? And assuming that they all have like a similar genotype, or they all have genes that would express green color, but some of them aren't expressing green color, is that an example of variable penetrance? Okay. So would we say it'd be both, right? So now some pedigree stuff, right? Well, it's not actually pedigrees, but it's related to a pedigree. Hi.
I have a question. So is it not incomplete dominance because there's two things like kind of in the middle, and you have some, like one particular like phenotype that can be considered like incomplete dominance? Because you know how like when it's red white and there's the pink phenotype, like I just can you explain like why this isn't incomplete dominance? I just like don't know because like there is like two phenotypes that are like and it's like similar, and then there's almost like I kind of categorize those two as like the phenotype in the middle.
Yeah. Okay. Yeah, that's a really good question, right? So, incomplete dominance, is this usually dealing with one or two different traits? It's in twos, right? Okay, so it's dealing with two different traits, right? So in incomplete dominance, we would see an intermediate phenotype in between two competing traits, right? So I was talking like red and white, like red being dominant, white being recessive. Let's say these are two different genes, each coding for some sort of color in whatever animal, right? So they're competing, and if it's incomplete dominance and you have a heterozygote, one with the red allele and one with the white allele, we're going to see something intermediate between red and white, right? But in this example here, we're speaking strictly about green, right? So we're not looking at like green and white, we're not looking at other things, right? We're just looking at green itself. And in this example, just one trait, right? Some turtles are green, some are not. So that would be the example of the penetrance. And then to what degree they express their green color, that would be our example of expressivity, if that helps.
So it's like because it's basically saying that it's not this gene being expressed as like the other. Okay, so. But if it was to be like, let's say, no green pigment was to be replaced with somehow like red, like a red turtle popped out, would that then be considered evidence?
If we had like two different genes for green and red, and we saw a red turtle. Okay, so that's a good question, right? So let's say we have two genes, one for green and one for this sake, let's say like a color that goes into green, so maybe like white. Yeah, so if we have, yeah.
I was thinking maybe it helped like for this example specifically for Grady up here. Like if we had turtles, tell me if I'm right, for codominance and they have like yellow and green spots, then you have codominance. But if you mix those two colors together and you have yellowish green or greenish yellow turtles, then you have incomplete dominance.
So the question says they then have no green. Yeah, so it's not expressed. Like it just isn't being expressed in those traits.
Yeah, maybe if it was like it has green pigment, you may be thinking like no pigment means like white pigment or something. So maybe if it actually said like it has green and white and now it's expressing like both discretely, then it would be like codominance, right? But in this case, we're not treating no pigment as an example of any color. It's just saying the absence of green, right?
Just the PowerPoint that like my professor had, it just said like incomplete dominance, like no pigment, no pigment. So this is
Yeah, that's not very clear. And yeah, another example, something that kind of fits into the mix here would be epistasis, and we'll get to epistasis a bit later. So 18, which forms of inheritance appear approximately equal in males and females? Okay, I got an autosomal dominant in the house and recessive already. How about any of the sex-linked traits? No, right, because they're sex-linked. I would say an autosomal means like a body gene. So now we'll look at which forms of inheritance skip generations, right? Holandric? Yeah, that's a good question. Does anyone know what holandric is? Your time to shine. Why? You're right, yeah. For skips, equally in males and females, we got autosomal recessive and autosomal dominant, right? Because autosomal traits are located on your body cells. They don't really care about your sex cells. And we all got the same body cells. We're all humans. So now, which forms of inheritance skip generations? So I heard something recessives. Yeah, so both recessives, right? So your autosomal recessive, sex-linked recessive. Because your holandric is not going to skip generations. So you have a holandric gene, you have a son, he's going to have it because you're going to pass, a male would pass his Y chromosome to his son, right? And his son would get his X chromosome from his dad. Mom. It's either or, mom or dad. So let's talk about like X-linked recessive genes, right? Or phenotypes. So would a male or a female be more likely to express an X-linked recessive? A male, right? Because there's a fancy word we're looking for, it's a vocabulary word. A male is hemizygous. It's a good word. So we got like heterozygous, we got homozygous, we also got hemizygous, right? That's meaning that since the male only has one X, right? he just needs one copy of the recessive allele to display the recessive trait. Right? The female would need two, obviously, because she could have one recessive allele, but there's also a chance she could have a normal allele. Right? So we'll look at number 20. Get a C, okay. So half recombinant, half recombinant, half non-recombinant. Got an E. You got an all recombinant with the parental phenotype ratio of 1 to 1, right? So let's explore these two letters, right? So let's look at C, right? Half recombinant, half non-recombinant, right? So I would say probably the key word here is recombinant, right? So how do we get recombinant gametes? Crossing over, right? And in the problem it states complete linkage, no crossing over. So it'd be unlikely we'd have recombinant offspring if there's no crossing over. And could anyone define what a complete linkage is? They're what? Appear together? Okay, awesome. Thank you for that. So this is saying that it's not like the cell didn't actually have the crossing over process happen, but the genes are so physically close together on the chromosome that the likelihood that crossing over would occur right in between those two genes is so low, we're saying they're completely linked. And they're pretty much never going to cross over and create recombinant gametes, right? So now, how about E? All recombinant with the parental phenotype ratio of 1 to 1. See, this question, it doesn't really specify here what the homozygote is, right? So that is quite misleading. I apologize about that. So in this case, we'll say it's a homozygous recessive, right? Because we know chapter 7 was all about test crosses. And we know test crosses are crossing a heterozygous individual with a homozygous recessive, right? This can be useful too if you have an organism displaying the dominant trait, right? You want to see if he's a heterozygote or a homozygote, you would cross him with a homozygous recessive, right? Because if he is heterozygous and you cross him with a homozygous recessive, you'll see in the next generation there will be progeny displaying the homozygous recessive trait, right? But if he was homozygous dominant and you cross it with a homozygous recessive, you're only going to get the dominant trait in the next generation. Yeah, so in this case, it would be E because there's no recombination occurring. And since we're crossing a heterozygote with a homozygous recessive individual, we would see a mix, right? So 50-50. And there's, I remember in her PowerPoints, she has like a whole section on different variations of like complete linkage, completely unlinked, kind of linked, and like the ratios in between, or how the progeny end up looking. So it'd probably be good for you guys to know the different patterns and like proportions of those different things, right? This is chapter 7. So now we're going to get more out of the inheritance portion of genetics, right? We'll dive into some more molecular stuff. So what are the components of a nucleotide monomer? And how would I differentiate DNA from RNA?
A monomer consists of a phosphate, a pentose sugar, and a nitron in its base, right?
Awesome.
And then RNA has a OH after the 2N, and DNA has only an H after the 2N.
Yes, thank you for that. So what she said, nucleotide monomer is a pentose 5-carbon sugar, right? Nitrogenous base attached to that sugar, and a phosphate also attached to the sugar. And we know ribose, deoxyribose. One is just ribose, but with deoxy in front of it, we can think of oxy as an oxygen, right? And so ribose would have an oxygen, an OH group, hydroxyl group, whatever you want to call it, at the 2' carbon, right? Deoxyribose would not have this oxygen. It would just have a hydrogen. And also, please note that the 2' hydroxyl on the ribose sugar is what makes it less stable than DNA. So that's why DNA is a bit better to serve as the holder of all of our genetic information, because it's more stable. We want to keep it stable. So now, how do we differentiate between a nucleotide and a nucleoside? The side does not have a phosphate, that is correct. Right, that was kind of confusing to me because I would hear people say like nucleoside triphosphate. I'm like, what are you talking about? That's the same thing as a nucleotide, right? So you're saying nucleoside triphosphate, I guess it may be better because when you're saying nucleoside triphosphate, you're indicating that there's 3 phosphate groups attached to the nucleoside. So yeah, nucleoside just has no phosphate group attached to it. It is just a sugar and a base. And if I was to say nucleoside triphosphate, it's pretty much a nucleotide, right? Because nucleotide isn't too specific about whether or not we have 1 phosphate, 3 phosphates, so. So now we'll look at which of the following statements is correct regarding DNA replication. So we'll look at D first, right? DNA is synthesized in the 5' to 3' direction on the lagging strand, but the 3 to 5' direction on the leading strand. How do we feel about that? See some head shaking. It's not good, right? DNA replication is the same direction, in both directions, right? Which is 5' to 3'. So is there one origin of replication in eukaryotic replication? No, there's a lot. There's a lot of origins. Are both strands synthesized in the direction of the replication fork? No, they're not. So what do we call the synthesized strand that's moving in the direction of the replication fork. So leading strand, and the other one would be the lagging strand, right? So our answer would probably be A, right? Only the new strands on the leading strand are synthesized in the same direction as the movement replication fork, right? So what's going to be different about DNA that's synthesized on the lagging strand compared to the leading strand?
It's going to be synthesized discontinuously.
Okay.
The lagging strand and synthesized continuously in the leading strand.
Yeah, so we have discontinuous synthesis, right? And what do we call these fragments? Okazaki fragments. And what about the primer situation, right? Because in the leading strand, we can just lay down one primer, and since it's moving with the fork, we're good, right? But what do we have to do about the lagging strand? Multiple primers, right? So we have multiple primers. We have the new DNA synthesized, but we have it's going primer DNA, right? And that primer is made of RNA, right? So we don't want RNA in our DNA, so we're going to have nicks in between these, in between the lagging strand, right? And that's when we'll need special enzymes to come in and repair the nicks and replace the RNA with DNA, right? So does anyone know what repairs the nicks? Ligase, nice. And what replaces RNA with DNA? Polymerase. DNA polymerase 1, right? What does DNA polymerase 3 do?
Has 5 to 3 polymerization and 3 to 5 hexanuclease.
Nice, yes, exactly. You said, what'd you say again?
5 to 3 polymerization and 3 to 5 hexanuclease.
So yeah, 5 to 3 polymerization, 3 to 5 prime exonuclease activity. Right. So exonuclease means to like rip off nucleotides, right? So if it's synthesizing DNA and it makes a mistake, it can go back in the 3 to 5 prime direction and remove that and place the correct one in. So, hi.
Hi, sorry. What answer did you set up for number 20?
Is E. Yeah. So what does the function of RNA depend on?
It's secondary structure.
Nice. Secondary structure. And there's 3 different like forms of secondary structure. Do you guys remember what they are? Makes those shapes, the RNA. The what? The types of secondary structures are. So we have like a stem. What else? Hairpin. We have a hairpin, right? And we have a loop. Nice. So stem, hairpin, and loop. These are different types of secondary structures we have in RNA. These are going to give it, this is going to give RNA its function, right? So we have a bunch of different types of RNA. So what's the central dogma of biology? Do what? The flow of genetic information, right? So what's the flow of genetic information? What is it flowing into? DNA to RNA to proteins, right? That's really good. So what's mediating the process of DNA to RNA? Transcription, right? What's mediating the process between RNA and proteins? Translation, right? So could someone just say what a gene is? A what? Unit of inheritance. Okay, cool. So what does a gene code for? Yeah, so it codes for a protein, right? And a gene's just that long nucleotide sequences. It's comprised of exons and introns, right? And that's what's getting transcribed. So it's just good to know what the whole point of all these genes are. So 26, right? Which enzyme is involved in DNA replication, or which enzyme involved in DNA replication has proofreading capabilities? B. What do you guys think? Does DNA polymerase 3 have proofreading activities? Could somebody like find it in the slides for me?
What's the question?
If DNA polymerase 3 has proofreading activity? So does DNA polymerase 1 have proofreading activity? It does, right? So the question is DNA polymerase 3. That's a good question. That's a good question. And it's saying it does have proofreading activity. Do you know what slide that is?
24 and chapter 12.
Awesome. Thank you so much for that. So yeah, chapter 12, slide 24, that's where you'll find the answer to that. But in short, I kind of messed up. It should have a choice for A and B, but it technically is both, right? DNA polymerase 1 and 3. And what does gyrase do? Can anyone tell me?
Relieves tension ahead of the replication fork.
Okay, good, yeah. So its other name is topoisomerase, right? And it does relieve supercoiling ahead of the replication fork, right? So which statement best describes the transcription apparatus most commonly seen in bacteria? Okay, thank you for that. Do you guys agree with C? Yeah, it's C, right. So no, 5 polypeptides and sigma factor. Not alpha, not beta, sigma. So which statement is incorrect regarding row-dependent determination? Do we know if a hairpin is formed at the RNA molecule? Rho dependent. Do what? Dependent. So you're saying there is a hairpin that's formed in rho dependent? Dependent. Independent.
I'm confused because I feel like B and D are both incorrect because I thought the rut site was upstream.
Yeah, so you are correct about that. The rut site is,
B and D.
well, you're correct about B being incorrect because the rut site is upstream of the termination sequence, right? It's not downstream.
I thought hairpin was independent.
Is that what?
I thought hairpin was formed in row independent. So that would be false. I thought row dependent would have to do with the rut site.
Are you seeing directly in her slides that there's no hairpin loop?
It says for row dependent, DNA sequences in terminator cause RNA polymerase to pause, rut site upstream of terminator binds to rut factor. Row follows RNA polymerase towards 3 ends, and then the helicase unwinds when it's reached. It doesn't say anything about hairpin, but it does in independent.
She put no hairpin?
Yeah, the picture showed a hairpin, but she said it was incorrect. So like,
So yeah, I messed up, guys. A hairpin is not formed,
So it's B and D for both of them.
so it would be B and D, yes. Good job. Yeah, yeah. B is in boy, yes, beta. You got it. So what about the function of rat1? Would it be B? Would you guys say that rat1 is involved in row independent termination? Seeing some shakes, hearing some no's, so no.
C.
Hearing a C. Operates in eukaryotic termination? Yes. Nice. Would anyone know how rat works? Anyone like to flex their knowledge? Right, yeah, so that's really good. So the pre-mRNA is cleaved when it should be cleaved appropriately, but then RNA polymerase keeps transcribing. So rat1 just latches onto the end of it and catches up to RNA polymerase and eats up the nucleotides it's transcribing, right? In a nutshell. So to what? Oh, hey, yeah.
Going back to 28,
So there is no hairpin formed in row dependent termination, but there is in independent termination. So how about that 5' cap? Does anyone know what it technically is? You're saying it's like ATG repetitive sequence? I don't believe so. Something about a methylated guanine nucleotide? Sounds about right. But anyway, that's not what the question's asking. Right? So does it increase tRNA stability? Seen. Do what? Yes, it's mRNA stability, right? Because T stands for transfer, M stands for messenger, and the transfer is going to transfer over the amino acid that properly corresponds to the mRNA's codon. So it does not increase tRNA stability. Is it involved in the transport of rRNA, mRNA, and tRNA? tRNA? No, right? It's just with mRNA, right? Because why do we want our mRNA to be stable? What's important about that? Yes, very good. And is it going somewhere? It's going out of the nucleus, right? Because genetic material is in the nucleus, ribosomes are in the cytoplasm. The nucleus is in like this pore, you know, it's got the envelope and such, so we need to transport this messenger through the cell, and when it's moving through the cell, there's a lot of stuff in the cell. We don't want it to interact with things in the cell. So we got to keep it stable, because if this mRNA somehow gets degraded, we don't want it to get degraded, that's a potential protein. That's a potential enzyme that you might need to break down food in your body or something, or trigger some immune response. So if it's not stable, you're not going to get the proper protein or enzyme, then bad things are going to happen, right? So that's why we take precautions to make sure this mRNA is stable enough to make it to the ribosome. But other than stability, the 5' cap also has another function. And it allows for the recognition of mRNA by the small ribosomal subunit, right? So it is involved in stability, but it is also involved in initiation of transcription. So this is a pretty basic question. Which type of enzyme functions in eukaryotic transcription? Just dealing with transcription, not replication. So is it RNA polymerase or DNA polymerase? I heard DNA. What is, and I hear RNA, right? So it can either be DNA or RNA, right? So where do we see DNA polymerase in? Replication, translation, transcription? Replication, right? Because DNA polymerase is catalyzing the polymerization of new DNA. And in transcription, we're making RNA, right? Replication, we're making DNA. Back to the central dogma, right? DNA, RNA to proteins. So now we know it's RNA polymerase. Now is it 1, 2, or 3? We know it's 2. Nice. So why do we consider the genetic code to be redundant? Does it contain more amino acids than there are codons? That wouldn't make sense, right? More codons than there are amino acids, right? So when you look at the codon table, you see like 5 or 6 different codons coding for one amino acid. That's what we mean by redundant, right? Because obviously it could not be, it contains more amino acids than there are codons because there would be some amino acids that can't be coded for. That would make sense. If you've taken a nutrition class, what I've just said might not be true, so ignore what I just said. So in eukaryotes, where is the start codon found? I heard a Kozak sequence.
Sorry, for 32, did you say it was B or D?
It is E.
32Z?
Yes. Do what? It's the promoter. What do you guys think about the promoter? Is that where the start codon's found in eukaryotes? Seeing a shake. Seeing a hand. Initiator sequence. What do you guys think about initiator sequence? Seeing another shake. Why do you shake? I think it's A. What do you guys think about A? Yeah, so shine, Dalgarno is in bacteria, right? Not in eukaryotes. So we can rule that one out. So it is A, right? Kozak sequence. So does anyone know anything about the promoter sequence? Is this actually like being coded, or is it in like the promoter region? Tell me about the promoter sequence. It's not transcribed? Okay. So we know the promoter sequence isn't transcribed, so it's probably in like the regulatory region of the gene, right? It's not in the actual coding region. And for looking at the start codon, we know AUG is usually start, right? And that's like methionine or something? Methionine. So if we know the start codon is actually going to code for an amino acid, then it's probably not going to be in the regulatory region of the gene that's not being transcribed, right? So IF3 has what function in translation? It's a P. So do you guys agree with IF3 facilitating the release of the completed polypeptide from the ribosome? Saw 2 shakes. C. So it is C, right? So IF3 will prevent the binding of the large and small ribosomal subunit during initiation. So we'll turn our attention to number 35. An operon related to an important enzyme has a regulator gene that encodes an inactive repressor. What type of operon is this? I heard a negative inducible and I heard a negative repressible. Right? So, I mean, they're both negatives, right? So we can probably assume it's negative. And the main indicator that it's negative being that we have a regulatory gene coding for a repressor, right? So negative or positive is just saying what the regulatory gene codes for, whether be it an activator or repressor. And something fun to note that regulator genes are actually coding a repressor or an activator, right? So like regulatory sequences that aren't in operons, you know, they usually aren't transcribed. They usually have like transcriptional activators or transcription factors bind to these sequences and initiate some sort of transcription. But in this case, the regulatory gene is a gene, so it's coding for a product, which is an activator or repressor. So. Okay, so we know it's negative, right? And we know the repressor is inactive. So if we have an inactive repressor, that means the repressor isn't actively repressing, so the gene's probably on, right? And if the gene is on before some sort of repressor or like substrate interacts with it, that means we can turn it off, right? So that means the gene's repressible, right? We have the ability to repress it since it's on, right? So do you guys agree with negative repressible? So it is negative repressible. So now 36, an operon related to a structural protein has a regulator gene that encodes an activator requiring an inducer before stimulating transcription. What type of operon is this? Heard some positive inducibles. I would agree with that. That's a good answer.
Positive because we have an activator, so that tells us it's positive. And inducible, meaning it can be induced. And when it can be induced, that means it'll be activated. So if we can induce it, it's probably going to be off, right? Because we're not going to turn something on that's already on. So it would be a positive inducible. So yeah, you know what type of regulatory gene it's going to be, positive or negative, if it codes a repressor or an activator. And if it's already on or already off, we'll tell you if it's inducible or repressible, right? So if it's inducible, like what I just said, it's going to be off because you can induce it to turn it on, right? And it's going to be repressible. That means the gene is repressible, meaning that it's going to be on initially, and when it interacts with a substrate, you can turn it off. Because you don't want to turn something off that's already off, or turn something on that's already on. So, catabolite, repression of the lac operon requires what?
Glucose was like the default metabolic pathway. So as long as it was producing AMP, you wouldn't have cyclic AMP. As long as it was producing ATP, you wouldn't have cyclic AMP around to initiate the other pathways, right?
You seem to be on the right track, yeah. So we know that cells like to utilize glucose as an energy source, right? Because glucose can directly enter glycolysis and create ATP, but cells can adapt, get around that. So they need to make ATP somehow, and they'll use any sugar they can really find. So if they can't find glucose, they'll take other measures to utilize other sugars and create ATP, right? So do we know the relationship between glucose and CAMP? Directly proportional, inversely proportional, inversely proportional, right? So if we have not a lot of glucose, we're gonna have a lot of CAMP, right? So does anyone know, so we'll probably need CAMP for catabolite repression, right? Does anyone know what else we would need? CAP? Okay, why? Okay, right, so they make a complex together, right? So we're going to need both of those things. Thank you, guys. CAMP and CAP. Repression?
Yeah, the catabolite. What was, because you said I was on the right track, but I still don't understand directly what it's referring to.
You're saying that if glucose levels are high, then you're not going to have a lot of CAMP, right? So I was just saying if glucose levels are low, you're going to have a lot of CAMP. And then a lot of CAMP will trigger catabolite repression when it combines with CAP, right? Low glucose will mean high CAMP. CAMP, yeah, inversely proportional. So, which cis-acting sequences in eukaryotes are positioned and orientation independent. So first, could someone describe to me what a cis-acting sequence is? Do what? They interact transregulatory proteins? Because what? Do you guys agree with that? These cis-regulatory, or these cis-acting sequences attract transregulatory proteins? You do what? Just explain what a cis-acting sequence is. Yeah. Yeah.
How was that its influence or silencer? That,
Okay, awesome. Thank you. So they control transcription. So the genes, they're on, right? So it's on the same gene. So then a trans would be then what? They bring in transcription promoters from other sources, right? So you think of it if you take in any organic chemistry or just chemistry in general, cis, they're on the same side, so they're on the same gene, right? Trans, opposite sides, they would have to recruit. So which of these cis-acting sequences are position and orientation independent? Promoters? Okay, do you guys agree that promoters are position and orientation independent? Excuse me? Dependent? Oh, I thought you were saying they were dependent. Wow. I put in my mind independent. I was reading that completely wrong. I'm sorry. So yeah, promoters are dependent, right? And we would know that enhancers and silencers are what? Independent of both, right? Sorry about that. So about epigenetics, right? It was promoters. So epigenetics, right? Can someone just say anything about epigenetics? So, say that again? Okay, cool. So we have like chromatin modification and sequences don't change, right? So this is a way of the cell to either increase transcription or decrease transcription without having to recruit transcriptional regulators, transcriptional activators, transcriptional factors, and you're not actually changing the DNA sequence, right? epi meaning on top of, right? So we're changing the structure of the chromatin on top of it by doing certain things to the chromatin or histones to control our levels of transcription.
Sorry to go back again. For 37%, it was, but I thought it was the opposite. Did you say E or did I hear you wrong?
Yeah.
But I thought that is for lack opera, like the activation of the lack opera. So what do we see? Right?
So can someone say what catabolite repression does? What is it repressing? What is it doing?
Isn't it repressing the transcription to make the proteins to like to deal with any sugar other than glucose? Does that make sense?
Yeah, so you're saying that it's repressing enzymes that break down glucose? Other sugars besides glucose. Could you, is that what it says on your slides?
Yeah. When you're not using glucose, you're using lactose. So it's repressing those proteins that lactoferrin codes for.
To break down glucose?
Yeah, lactose.
To break down lactose.
It's repressed expression of proteins needed for metabolism of other sugars, other than glucose. Yeah, but I'm just looking at the slide. Because it says, okay, so when you have high glucose, you're going to have low levels of CAMP and CAP, right? And when you have low levels, you wouldn't need a lactoferrin because you have glucose. When you don't have glucose, then it produces CAMP, and that'll turn it on. That'll turn on the lactoferrin to use lactose instead of use the other ones instead of glucose. So you're doing the opposite. Activation. Yeah.
Activation to suppress, to not, to use glucose, right?
But the question, the other question is saying it's suppressing. I think they're saying it's suppressing other things so they can.
Huh?
Glucose is present at lactoferrin expression. Correct? Yeah. Yeah, exactly. Yeah. So if the lactoferrin is on, then it codes for those proteins that allow the cell to use other sugars besides glucose, like lactose. Yeah. Yeah.
Yeah.
So then, 37 would be C, right? Well,
No, because the lactoferrin would be on.
if there was none. But you're right. If there's glucose, the lactoferrin is going to show. But
So it wouldn't be.
Like
you're
bring-
repressing it. Is the question asking what do you need to repress? Repress it? Because when
second. So,
I just think it's C.
In the group me, the grad TA said that none of those answers are correct. He
really?
said that David got confused on 37s. CAMP and CAP are involved in activation. And I said, so what's the answer? And he said, no answer works here. Lactose will express.
Well, there you have it, folks.
So what is it? Is it lactose and allolactose then? Because it says if lactose and allolactose are present, the repressor is inactivated, and RNA polymerase is not blocked, and transcription occurs.
So,
Because allolactose is needed as the inducer because isn't it a negative inducible operon, so you need something to activate it, and allolactose is acting as the inducer, so you're going to need allolactose.
Well, when you have lactose glucose, you get cyclic AMP, and then cyclic AMP,
Those are activators, yeah.
yeah, that's the activator, and that creates more of your
I would disregard the question. I'm sorry about the confusion it brought. I'm sorry if I misinformed you. I misinformed myself. So, yeah.
Yeah, I know that. Is the inducer.
Yeah, so hearing some different things. Sorry about all this. We'll just move forward. No, I'm sorry about all the confusion.
I think it's explained better in the textbook. I read the textbook.
Yeah. So the textbook is a great resource.
Slides are confusing to me, so I reread it.
So, yeah, great discussion, you guys. Thank you for not just listening to me and nodding and thinking individually and independently. Props to you guys. So now we're going to talk about what modifications can be made to histone proteins, what effect these modifications have. That's good, that's good. So yeah, acetylation, it's a really good one. And what does acetylation have? What's its effect? Right? Yeah. So histone acetylation will increase the positive charge and effectively increase transcription, right? Because it decondenses the chromatin. Because as we know, DNA is negative, histones are positive, and DNA is tightly bound around the nucleosome. And it's tightly bound, we can't get in there, enzymes can't get in there to transcribe, so we need something like acetyl groups to come in and loosen up this condensation. And okay, so can methylation occur to histones then? Can you methylate a histone? So remember something about like CG islands or something? CPG islands. That's saying that the methylation is going to occur somewhere in that CPG island, right? And that CPG island is located where? On the nucleotide sequence, right? So could we really methylate a histone?
Yeah. In the slides, it literally says histones can be methylated.
Yeah, it does.
So we can methylate histones. And methylation, she kind of says like two different things. It can like repress or suppress, or repress or activate, but she mainly gives like examples of methylation repressing. So I'll probably just know specifically, it's usually repressing, but it can, in some cases, activate. So what attracts chromatin modifying proteins to restore chromatin modification on newly synthesized DNA? So we just had our methylated DNA replicated, right? And as we know, replication is semi-conservative. So after replication, we'll have two new strands, half parental, half new, and the parental strands will have methylation patterns on them, right? And we need the methylation to occur on the newly synthesized strands too, right? So we need something to recruit these chromatin modifying proteins. Yes? It is. Very nice job. Hemimethylated DNA. Hemi meaning like half, because the new DNA is going to be half acetylated, half newly synthesized, so it's not acetylated, methylated. So yes, the hemimethylated DNA will attract these chromatin modifying proteins. One of them, for example, could be maintenance methyltransferase, right? So what in eukaryotic DNA allows mismatch repair in a cell to distinguish between the old and the new strands of DNA? It is the nicks in the strand, right? Because that's the one way you can distinguish between the new strand and the old strand. Because if the cell doesn't know which strand's the new and the old, it needs to preserve the old, right? It doesn't want to say, oh, the old is actually the mismatched one, then change the old strand, and then you have a completely different sequence, right? So the nicks and the new strand are good indicators for the cell to distinguish between new and old strands. Yes. Yes. Say that again? When we're speaking about methylation, I was speaking about like the attraction of chromatin modifying proteins in epigenetics, right? But in this case, we're speaking of like an accident DNA replication or something. And how would the cell know that which strand's new, or which strand is the one that has a mistake on it, right?
For bacteria, the old strand is the one that gets methylated, right? That's how you distinguish the difference.
It does what?
The old strand is methylated, so we know the difference between the new and the old one.
I thought the old one is
Yeah.
Yeah. Oh, that's,
Yeah.
sorry.
Say that one more time.
For just the same thing, but like in bacteria, is the old strand is the one that's methylated. That's how we know the difference.
Yeah. So what steps do we take to turn free mRNA to mature mRNA? Five prime cap, poly A tail. All very good things. One more. you splice these things out. Introns, right? Because as we know, introns are non-coding sequences. We don't want introns. And having introns also opens up the window to alternative splicing, meaning that we can excise some introns and different exons out of the mRNA to create different proteins from the same sequence. All right, last but not least, epistasis. So could someone describe to me what a recessive epistatic gene is? Yeah, that's good. So that's when a recessive allele would mask the phenotypic outcome of other things, right? So if I was to write, right, so B could code for some color, and so could E, right? And when E would be dominant, they could maybe combine their colors, or you'd have a dihybrid cross where it'd be tall and blue, tall and white, short and blue, short and white, right? But when you have two little e's, since it's epistatic, it's going to mask the expression of the b's, right? So no matter what we have in the b's, that would usually indicate like it would have some influence on the color, the e is going to dominate overall. So the two little e's doesn't care about what the b's are saying. It's just going to say, the organism is going to express whatever the little e's are expressing without regard to the b's, right? So that is it for my exam review that had a few mistakes in it. Thank you guys for showing me what I did wrong. Really appreciate it. And thank you guys for coming here. Does anyone have any questions about anything that we could talk about? I don't know anything about the final exam. Yes. Okay. For this? Yeah. Yeah, of course. So one would be A. Yeah. 2, it's kind of like free response. Yeah, so diploid means like you have, yeah, 2 and N. Yeah. Yes, sir. G1, G2. 4 is D, 32. 5 is after anaphase. N equals 2 to the N, where N equals number of homologous pairs. 6 is D. Oh, sorry. Oh, yeah, that was 6. That was 6. Yes, sir. 7 is D. 9 is also D. And 10 is B. Yeah. And, yeah, 8. Recombination between non-sister chromatids does not create new variation. Does not. Mutation does.